Whoa, whoa, whoa. Welcome to topic 4, the structure and function of RNA. RNA is a nucleotide just like DNA, and so we're going to see how they're both similar to each other on this slide. So they both have nucleotide monomers, and as a reminder, nucleotides compose of sugar, phosphate, and a nitrogenous base. Now, those nucleotides are connected by a covalent bond called the phosphodiester bond, and it links it together with the 3' and 5' end of the nucleotides, with the nitrogenous bases perpendicular to the sugar phosphate backbone. So DNA is usually a double-stranded molecule made up of nucleotides that contain the sugar deoxyribose, while RNA is usually a single-stranded molecule, can be double-stranded in viruses, made up of nucleotides that contain the pentose sugar ribose. So you don't need to know the big difference between the ribose and deoxyribose structure, but just as a note, it's on the 2' end, y'all. Difference is ribose has a hydroxyl group, while deoxyribose has a hydrogen. So again, they have nitrogenous bases, right? Pyrimidines and purines. So we went over on how to memorize those structures on the previous lecture. So DNA contains TAGC, and then RNA does not contain thymine, where thymine is, it is uracil. So it has UAGC. So again, complementary base pairing still applies. Cytosine always pairs with guanine, and then adenine pairs with thymine and DNA, or uracil and RNA. So here's just the structure of uracil here. We can see that it is a pyrimidine with a one-ring structure, just like the rest of them. And then purines, they have two-ring structures. And so this again goes with the anti-parallel concept. 5' end has the phosphate group, 3' end has the hydroxyl group. There are many different types of RNA molecules found in living organisms, and they all have different functions. The biggest one we're going to cover in detail is protein synthesis and gene expression in the next lecture. Those involve messenger RNAs, ribosomal RNAs, and transfer RNAs. Then we have microRNAs, and they are involved in regulating the gene expression, meaning what helps control how genes are turned on to be expressed or turned off. We have ribozymes, the enzymes made of RNA molecules that catalyze specific chemical reactions. So just to reduce that activation energy to make it go faster. So last but not least, y'all, RNA is genetic material in many types of viruses, like mentioned before. They can be double-stranded in viruses, just like DNA is for us. So the RNA is definitely responsible for the storage and transmission of their heritable information. Okay, RNA is that as well as DNA. Again, there are three main types of RNA, and they're found in all cells: rRNA, mRNA, and tRNA. With the rRNA, they're molecules that bind with proteins to create ribosomes. Each ribosome is made up of two pieces, y'all. You guys can see it here. They're the small subunit and the large subunit. Again, they combine together during translation to create that polypeptide chain. So, ribosomes are the site of protein synthesis. mRNA copies the genetic code from DNA and brings that information to ribosomes. So the DNA, again, is too big to leave the nucleus, and it has a lot of information. So not all of the DNA needs to be translated. So mRNA is there to, again, carry a specific portion of that specific protein that's needed. to be made, and it brings it to the ribosome. So on the messenger RNA, there's something called the codon. The codon is just a series of 3 letters here, then nucleotides, right? So each codon specifies a particular amino acid, which can be determined using a codon chart. Now, you guys can see it here very clearly. So again, we have a very specific amino acid called lysine, and it's called upon by AAA. Okay, and so there we go. So again, this is the codon chart previously mentioned, and I'm going to use the example of the codon AAA to verify that it is indeed lysine. So for the first letter, we need to take a look at the left-hand side. I isolated A, so I'm going to highlight this in green. Then for my second letter, I have A, so I am isolating this section here in blue. And finally, for my last letter, I'm going to be concentrating on this specific line. So right here. And then that tells me that this is indeed lysine. And then that's how you read the codon chart. Last but not least, tRNA. They're RNA molecules that bring amino acids to the ribosome so that the polypeptide chain can be built. Each tRNA binds specifically to an amino acid. And then it contains something called the anticodon. So the anticodon is a sequence of 3 nucleotides that is complementary to the codon on the mRNA. So let's take a look at the structure of tRNA. We have the amino acid located here. The RNA runs from 3' to 5' in this image. So we know that the anticodon is 3' to 5'. The anticodon then again goes to the codon and complements it. So notice that the mRNA, the codon, is being read by the ribosome from the 5' to 3' direction. So again, the 3' to 5' is complementary once we see and reverse the structure there. Okay, so again, it complements the mRNA. Okay, there we have it, y'all. Have a good one.